So in last part, we have discussed the genotypic proportion or gene frequency in a population which is under random mating. Now we will prove a theorem in which we want to prove that the genotypic array of a population produced by random mating is the square of gametic array in the initial population. So first of all, I want to introduce what is genotypic array and what is the gametic array. Let us start with the initial population with two alleles at a locus, say A. So if mating takes place between these alleles, so only one type of genotype we will produce. And suppose P is the frequency of allele capital A and Q is the frequency of allele small a in such a way that P plus Q is equal to 1. Now from these two alleles we can get one type of individual, that is capital A small a. If the crossing of capital A small a is with crossing of same individual, means selfing is there, then in resulting population or resulting genotype, we will get capital A, capital A, capital A, and small a, with their gene or genotypic frequency p square, 2pq, and q square. Right? So this is my initial population when the gene frequencies are p and q, and this is my population in offspring, in which in initial population only one type of individual we have with two allele A and small a with proportion P and Q. So the gametic array of initial population is P A plus Q A, and the genotypic array in next generation will be equal to P square AA plus 2PQ small a plus Q square small a. So this is the genotypic array in next generation. Now come to the theorem. First we consider the initial population. We consider a population with G different genotypes, say G1, G2 and GG. The proportion of each genotype in the population is say P1, P2 and PG in such a way that sum of all the proportion I vary from 1 to G, Pi will be equal to 1. Now each genotype Gi produces a set of gametes, say alpha 1, alpha 2 and alpha s with specific proportions, say Pi1, Pi2 and PIS. Now the random mating takes place. So the mating between two genotypes, that is GJ occurs with probability, since the probability proportion of GI is PI and proportion of GJ is PJ, and it is independent. So we just multiply these proportion in order to get the probability of their matings. Means GI, if it is crossed with GJ, then the proportion I will get PI PJ. So when GI gametes mates with GJ, their gametes alpha M from GI and alpha N from GJ, they combine, but their proportion are say PIM for alpha M and PJN for alpha N. So when these gametes fuse with each other, so the genotype produced will be alpha M, alpha N, and their proportion will be PIM, PJN. Now talk about genotypic array in the offspring. Genotypic array in the offspring. The total contribution of a specific genotype, that is, we have produced alpha M, alpha N genotype, is the sum of contribution from all possible methods. And this will be given by expression I, J, Pi, Pj is the contribution of this mating Gij and the sum over M and N, since the genotypic array or gametic array of Gi is having M gametes and Gj has N gametes. So their sum will be PIM, PJN, alpha M and alpha N. If I simplify this, so written as this can be written as this expression. Means I take this summation sign M and N outside and the two summation over I where Pi, PIM, alpha M. Since they are independent of each other, so this can be written as J, Pj, Pjn, alpha N. So now let us understand it, how this occurs. The term Pi, PIM, alpha M represents the contribution of genotype Gi. The gametes alpha M in the initial population. And essentially, it tells us the frequency of gamete alpha M in the population. Similarly, the second term Pj and alpha N represent the frequency of the gamete alpha N. So I think this is clear to you. Now combine the contribution. So this expression, summation over M and N, then summation over I, Pi, PIM, alpha M, multiply with summation over J, Pj, Pjm, alpha N, combine the contribution of all pairs of the gamete, say alpha M, alpha N. Now we will factorize it. You will notice that this term appears twice, once for each gamete alpha M and alpha N. This is because we are considering all possible combination of gametes from the initial population. So this sum over M and N is distributed for these two terms like this. So summation over M multiply by summation over I, PIM and alpha M multiply by N So this summation sign, the double summation is distributed over these two terms, this term and this term. Since these sums over m and n are independent, we can combine them in a single sum like this. So you can combine them in a single cell. When you combine, so this thing is same as this thing, so we make it square. So we can conclude from this theorem the expression m summation I, pi, m and alpha m represent the gametic array of the initial population. And by squaring this sum, we account for all possible combination of gametes, which gives us the genotypic array in the offspring population. So you can say the genotypic array of a population is the square of gametic array in the initial population. So let us discuss some application of this theorem. Again, let us consider an application of this theorem, that the genotypic array of population produced by a random mating is the square of the gametic array in the initial population. So let us create or let us consider initial population, which consists of three genotypes, say capital A, capital A, capital A small a, and small a small a. There P is the proportion of capital A individuals, 2Q is the proportion of capital A small a individual, and R is the proportion of small a individual. So the contribution of allele A. What is the contribution of allele A? Can be obtained by this term and this term. Here is the whole contribution. Both allele contributed to P, and here one allele contributed to 2Q and one small a contributed to 2Q. So the contribution A can be found out by P plus taking half of this heterozygous proportion. So P plus Q. And contribution of the small a can be obtained from R and half of this. So this is R plus Q. Now you find the contribution of allele A and small a. So we can form the gametic array. Gametic array can be obtained by this expression, P plus Q capital A plus R plus Q small a. So according to this theorem, the genotypic array in next generation will be the square of this gametic array. So let us square it. So what we do is P plus Q whole square as A plus, or you can say Q plus R small a. If I square this gametic array, what will be the result? So I will get P plus Q whole square capital A plus twice P plus Q and Q plus R capital A small a plus this is Q plus R whole square small a small a. Now let us simplify it. What we will, so these are the genotypic frequency in the next generation. So this theorem also indicates that under random mating, a population reaches equilibrium after one generation. Whether it reaches in equilibrium or not, we confirm that we will get the same gametic array as we get in the initial population. So this is the genotypic array in next population. Now we will find out the gametic array after one round of mating. How you will find out? Now you will see the genotype we have capital A, capital A, capital A, and small a. With their proportion, you can say P plus Q whole square plus half of this. So half of this will be equal to P plus Q into Q plus R, and this is the you can say proportion of gene or allele A. Similarly, you can find out the proportion of allele small a by taking Q plus R whole square plus half of this. So it is P plus Q plus into Q plus R. So you will get this expression. So this is the gametic array of second generation. If I want to simplify this, P plus Q square, P plus Q and Q plus R. If I simplify this term first, so P plus Q whole square plus I just multiply these terms. This is P into Q plus no, first of all I have to take something common. Now from this term you will see P plus Q can be taken outside. So P plus Q I take common and I will get P plus Q plus Q plus R, and it is P plus Q, this is P plus 2Q plus R, and this terms is equal to 1. So I will ultimately get P plus Q. Similarly for allele A, this is Q plus R I take common and I will get Q plus R plus P plus Q, and it is Q plus R, and inside the bracket we have P plus 2Q plus R, and this is again 1. So ultimately the gametic array I will get is P plus Q into A plus Q plus R into small a, which is same as that of the initial population, which is again same as the initial population. So this gametic array is same as we started with, which indicated that the population is in equilibrium. The second generation is identical in structure to the first, which confirms that population has attained equilibrium after one generation of random mating. And this application shows that any population will reach equilibrium for a single locus after one generation of random, and the genotypic array remains stable in subsequent generations also. The process and result described align with the theorem's implication, making this is a valid application of the theorem. And of course, there are certain consequences of the theorem in particular cases. What happens if we take two allele? What happens if we take three allele? What happens if we consider m allele at a locus? And so on and so forth. So we will discuss all these things in case of two allele, which is known as simple Mendelian inheritance. So if we consider a population with two allele capital A and small a, so these are my two allele, and P is the frequency of allele A and Q is the frequency of allele small a. It is given that P plus Q is equal to 1. Then the genotypic proportion. If random mating take place, three types of genotypes are produced with proportion say P square 2PQ and Q square. Suppose this P square is represented by capital P and this is represented by 2Q and this is represented by R, then what will be the genotypic array in next generation? In first generation, initial generation, the genotypic array will be PA plus QA. This is the gametic array, you can say, of initial generation. So, by using the theorem, the square of the gametic array will be the genotypic array of genotype in next generation. So, we just square it. PA plus QA, its square. So, on squaring this, we will get P square capital A plus 2PQ capital A small a plus Q square small a small a. So when I square the gametic array, I will get this expression. And this is well known Hardy-Weinberg equilibrium, which we will discuss later on in next class. Now here, the proportion of homozygous dominant individual. These are homozygous dominant individual. And what are its proportion? Is say P square. And proportion of heterozygotes is 2PQ. And proportion of homozygous recessive individuals is say Q. Now what are the implications? The implication of two allele at a locus, the population is in equilibrium under random mating, meaning the allele and genotype frequency remain constant from generation to generation, provided no other forces such as selection, mutation, and migration act on the population. Now come to case of three alleles. Suppose we have a population with three alleles, capital A, B, and small a. P is the frequency of A, Q is the frequency of B, and R is the frequency of allele small a, in such a way that P plus Q plus R is equal to 1. Now, what is the genotypic frequency of capital A capital B, R, R. So genotypic frequency in the initial population are say P square, this is Q square, and this is R square. Of course, when three individuals are mated with each other, so we have various types of, you can say six type of individuals are produced. Means capital A, capital B, small r, and then we have capital A, capital B, small a, and last one is A and B. So six type of individuals are produced. So their genotypic frequency in next generation will be P square, Q square, and this is R square. This is 2PR, this is 2QR, and this is 2P. So what will be the genotypic array in the next generation? We can find it with the help of gametic array in the initial generation by squaring it. So PA plus QB plus R, or you can say RA, if I take, because this is the gametic array in the initial generation. So I just square it. What I will get? I will get P square AA plus Q square BB plus R square small a plus twice P R capital A small a plus twice Q R B small a plus twice P Q. So you will get, and I just put A B also. So this is the equilibrium condition for a population with three alleles which are under random mating. Now, implication of this result, the allele and genotypic frequency will stabilize again and remain constant in subsequent generation, which demonstrate that the population has reached in equilibrium after one generation of random. We can generalize this result for M allele. So suppose you have A1, A2 and AM alleles with frequency say P1, P2 and PM, initial frequency, with the condition that the P1 plus P2 plus so and so forth PM will be equal to 1. The gametic array or genotypic array, you can say, in the initial population, we will get P1 A1 plus P2 A2 plus plus PM AN. So this is the gametic array in the initial population. Now we want to find out the genotypic array under random mating in next generation, which can be obtained by just squaring this. And when we square this, you will get an expression like this. So it is P1 square A1 A1 plus P2 square A2 A2 plus Pm square Am Am plus the cross product terms, twice the cross product term, means if it is P1, P2, A1, A2 plus so and so forth. So symbolically, this can be represented under summation sign, summation over I. So this is Pi square AI AI means A1 A1, A2 A2, AM AM plus summation where I is less than J. It is Pi Pj AI AJ. And this expression is in equilibrium. So in this expression, we will get the Pi square AI AI is the frequency of say homozygotes for allele AI and twice Pi Pj AI AJ is the frequency of heterozygote for allele AI and AJ. So we can find out its implication. The population is in equilibrium under random mating for any number of allele with the genotypic frequency being determined by the square of the gametic array. So you can find out the genotypic frequency of any random mating population in next generation by just squaring the gametic array in the previous generation. So you can generalize this theorem. The theorem can be generalized to consider the genotypic array of the offspring population under random mating when male and female have different gametic array. So up to now we assume that male and female contribute same. But if they have different gametic array in case of sex linked gene, so you can take example of gametic array of male and gametic array of female. So under random mating, the genotypic array of the offspring is equal to the product. In this case, what we can find out? In previous case, suppose PA and QA is my gametic array in initial generation. So the genotypic array of the offspring can be obtained by just squaring this. But if male and female may have different gametic array, then how you can find out the genotypic array of offspring in next generation? That can be just multiply their gametic array of male as well as female. Let us consider an example. Suppose the male population has gametic array say PAA plus QBB plus RAA. This is the gametic array of male. And suppose PA dash A plus QB dash B plus RA dash A is the gametic array of female. Then the genotypic array of offspring can be obtained by just multiplying this PAA plus QBB plus RAA with the gametic array of female PA dash A and QB dash B plus R A dash A. So by this product, you can find out the genotypic array of the offspring if the two parents have not same gametic array in the initial population. So we can conclude that the consequences and generalization of theorem are fundamentals for understanding how allele and genotypic frequency behaves in the population under random mating conditions. They are very important in field of genetical statistics, population genetics, evolutionary biology,